So welcome to the DevoWorm meeting for today. Since we didn't meet last week, today I wanted to give an update on the final submissions for Google Summer of Code. Our two scholars, Jayadratha, Gayen, and Lalith. And then I wanted to go over a few papers that are in our reading queue. So the first thing I want to talk about today is congratulations to our Google Summer of Code scholars for this summer. Jayadratha, Gayen, and Lalith, and they've completed their projects and committed them successfully to the DevoGraph repository in DevoLearn. So this is our DevoLearn organization here. We've been working on this for the past 7 or 8 years now. The software for analyzing microscopy images, analyzing other types of developmental data, and then developing algorithms, deep learning algorithms, and more recently, graph neural networks to analyze those data and turn them into visualizations and theoretical insights. Jaya and Lalith have been contributing to DevoGraph. This is the DevoGraph repository. The contributors section needs to be updated to reflect the recent gains in the content. The first one is DevoTG. This is the one that Jayadratha worked on this summer. This is DevoTG stands for Developmental Temporal Graph Networks. So these are temporal graph networks, a subset of GNNs, where you look at the time dependency of different network topologies and how they change over time. And so this was the project where he was using a secondary data set to calibrate these graphs and to build visualizations. So he got a lot of work done this summer. There's a lot of code in this repository. You have, of course, the data set that he used. He has the cell lineage data set, a connectome data set, and then some sample data here. So this is the relationship between a parent cell and two daughter cells, the birth time given in minutes of development, the position of the parent given in XYZ coordinates, and then there's this mapping between the parent cell and the daughter cell, so you can follow this through the graph. So this developmental lineage tree is growing through time. It's growing, branching downward. And then, of course, you have the connectome data set, which is the connectome cells. So he used the connectome data from WITVLEET 2021. The WITVLEET 2021 data set is a synaptic data set, so it looks at the synaptic connections between cells or the chemical connections between cells. And those data are available for 4 stages of larval development as well as the adult. So he's actually able to get developmental data for C. elegans. So you're not just dealing with more or less a static adult connectome, but the larval connectome as well. So this developmental data set involves 8 developmental time points. And in our meetings from this summer, Jaya was showing some of the visualizations of these different graphs. And so I don't know if he quite got to a point where he synthesized all of these developmental graphs into one visualization, but definitely that's maybe a follow-up part of the project. So the idea of these projects is just to get as far as you can over the allotted course of time. So there are many things that probably can be done with these data and these visualizations in the code. So it looks like if we walk through this, if you go through, he has everything in Colab notebooks, so you can run them from the notebooks. If you clone the repository, you should be able to run the notebooks without too much problem. The first point is the cell division analysis, so you start off there. Then you have the connectome development analysis. All the data should be included in these CSV files. Then you complete your analysis pipeline. So this will download connectome datasets, process them into temporal graphs, perform the network analysis, generate comprehensive visualizations, and create interactive animations. So this is a cell lineage data set. We have the parent cell, the daughter cells, as we saw before, on the position with the birth time. And then we have the connectome data sets, and we have 8 connectome data sets. And so if we take a look at the data, we see that there's this presynaptic cell, this postsynaptic cell. So remember, we're talking about the mature neurons in the connectome. So we have a different nomenclature than we did for the developmental cells. We have ADAL and ADAR, which are ADA left and ADA right. So those are right-left pairs. So there's a presynaptic left partner and a postsynaptic right partner, which suggests the directionality. You actually have in this connectome, you have both electrical and chemical synapses. So it actually includes electrical synapses as well. And so then this just number is just the type of synapse. So if we have all of these, we can actually look at the chemical synapses. We can also look at the electrical connections, and we can get a sense of what that connectome looks like. But we have these data over larval development. So these connections are going to change across larval development. And then within this data set, there are also counts. If you go through and analyze it, there are counts of synapses per pair. And so these are the notebooks. And just look at some of the visualizations. This is the division pattern analysis. So we ran a nice analysis on this. You ran a generation analysis, which is here. So we have a number of box plots and scatter plots. Here's the time progression analysis. So we have these point clouds that show the position of cells and the X and Y coordinates. And then for the connectome analysis specifically, we have this evolution of connection types over development with electrical synapses and chemical synapses. So the majority of these in the data set are chemical synapses. Then he has these network growth metrics, and these just show the growth of the network over time. Then finally, this is the network evolution visualization. So this just shows these networks as they change over time. So he was able to show kind of this change over time for the 8 different topologies and how those connections change. So if I go over it again, you can see that there changes over time as the network evolves. And so there's a lot of information to be gained out of these networks and how they change over time. You can gain statistical information, but there's probably also a lot of topological information that we can extract as well. So congratulations to Jayadratha on a job well done on this DevoTG project. Now Lalith did the NDP-HNN project, which stands for the NDP is this neural developmental programs, and then HNN is hypergraph neural networks. So the neural developmental programs approach, we used this last summer. We would develop that to the point where we could actually apply it to C. elegans, and then Lalith picked up that and carried it forward to the work he's doing now with hypergraph neural networks. So basically, the 2 projects last summer, the hypergraphs and neural developmental programs, were combined into this project. So Paki Vanchalia did the work on neural developmental programs. So thanks to her work, he's able to move this forward. So this is his repository. Looks like you can set the environment up. You can clone the repository, set the environment up. You have to specify your data format. This is the way he has the data set up. So he has different parameters like parent cell, birth time, and then there are these different parameters that are passed in, and they're a certain data type, and then he gives notes on that. So this is the documentation here for a lot of basically the idea is we're going to have different data sets that we can import here and use them as needed. So it gives instructions on how to run things and then troubleshooting as well. So the way Lalith has put this into the GitHub repository, he has different PY files, and so he doesn't have the notebooks, but he has the PY files, and you can run this in an environment or compile it if you want. So he has the code for generating hyperedges. He has the code for loss function. He has the code for the HNN model. And then he has the code for visualizing the outputs in UMAP or k-means clustering. So if you're interested in knowing more about his project, he's given a number of different presentations on it over the course of the summer in the DevoWorm meetings, so I refer you to those meetings to see how some of this code works. Congratulations to Lalith on a job well done, and good luck with both scholars in the future. So the first paper for today is this paper, The Regulatory Landscapes of Neurite Development in C. elegans. And so this is a little bit different way of thinking about the connectome. The abstract reads, neuronal communication requires precise connectivity of neurite projections, and examples are axons and dendrites. Developing neurites express cell surface receptors that interpret extracellular cues to enable correct guidance towards and connection with target cells. So this is talking about how when you have your lineage tree, those cells, some of those cells eventually become neurons. And in the process of becoming neurons, they have to connect to other neurons. And so neurites are these projections or these things that come out of the cells as they're differentiating and connecting that guide the connections, the axonal connections to other cells. So axonal connections, of course, enable these chemical connections that we talked about in the Whipfleet data set and enable the connectivity of the connectome. So we have the electrical connectome, which are these gap junctions where the cells come into contact, and then you have these projections, these more formal axonal connections, or these axonal projections that allow for these synaptic connections to be made. And so that's what they're looking at here. So developing neurites express cell surface receptors that interpret extracellular cues. So you have this gradient that exists within the developing nervous system that guides these connections towards the correct partner. As you noticed from the data set, there are certain cells that are connected with other cells, and those connections don't radically change across different organisms. When you have plasticity, there are specific connections made that are functional. And so you have the ability to trace circuits within the C. elegans connectome. And part of that is due to this system of axonal guidance that axons can make to the correct partner in the nervous system to make that connection and complete that circuit. So that's what they're talking about here. And this is a little bit different view on the connectome because it fills in the gaps of our developmental connectome. So one thing we have, of course, is the lineage tree. Another part is this ability to look at the plasticity, both chemical and electrical synapses. And then the third part then is how do you get this structure form from just basically differentiating cells? And that's what we're doing here. Spatiotemporal regulation of neurite guidance molecule expression by transcription factors is critical for nervous system development and function. So we have this, of course, this set of transcription factors that are activated in different cells, and this is spatiotemporally distinct. So a neuron or a developing neuron in one part of the nervous system is going to have a different set of cues than a cell in another part of the developing nervous system. And this gradient also exists in spatiotemporal space or spatiotemporal context, where you can have this gradient between two cells. The neurites know how to guide the axon to the proper target and can distinguish between targets. If you notice from the data set, at least in C. elegans, these cells are generally in a certain place in the anatomy, and so that's kind of playing into this whole model. Here we review how neurite development is regulated by transcription factors in the C. elegans nervous system. By collecting publicly available transcriptome and chip sequencing data, we reveal gene expression dynamics during neurite development. And so chip sequencing allows us to look at epigenetic phenomena, how genes are expressed. And then, of course, we have our transcriptome data, which is actually a measure of mRNA in like a certain volume of tissue. This provides insight into transcriptional mechanisms governing construction of the nervous system architecture. So this introduction just talks about sort of how animal nervous systems have developed to enable the transmission of information over long distances. Information transfer between neurons relies on neurite projections called axons and dendrites. So axons and dendrites, axons are going out from a cell, dendrites are coming into a cell, and they allow for information processing to occur. And so the neurites are basically these projections from the cell body itself. Correct neurite development is therefore critical for efficient signal transduction between neurons within a neural circuit. During development, neurites are guided to their targets by attractive and repulsive cues from surrounding cells and tissues and the extracellular matrix. So the extracellular matrix is the space between cells where the cells are migrating within, and this is where these gradients are set up. This is where these projections are being made, and this is allowing for this sort of connectivity. And you have these basic, what we call taxis. So we see taxis at the organismal level in C. elegans. But we also see these taxis at the intracellular level in C. elegans. And this is an example of a taxis where these neurites are sort of guiding along a gradient. They're being repulsed and attracted to certain chemicals, certain chemical concentrations, and it leads them to the proper target. Otherwise, in an environment where they didn't have those cues, they would just kind of branch off at random, and connectivity would just not be possible. You might get some connections by chance, but most of the development of a coherent connectome would not occur. And so this whole idea of neurite guidance, we need to identify ligands and receptors. So why are we interested in gene expression? Well, a lot of neuron subtypes, especially within C. elegans, where they're determined deterministically, have specific neurite projection patterns that are guided by distinct gene expression programs. So we know kind of that cells are going to be in a certain place, and the gene expression programs are activated at a certain point in development. And of course, neurite development requires precisely controlled spatiotemporal expression of these factors. And so transcription factors are key regulators of gene expression that control multiple processes of neuronal development, including polarization, migration, and guidance of the neurite projections. So a number of things need to happen in order for this process to be accurate, and so those are all set up by transcription factors. Transcription factors regulate neurite development by controlling signaling, cell adhesion, and junction molecules, as well as cytoskeleton modifiers. So transcription factors really do play a key role in regulating neurite development and their coordinated expression in time and space. Transcription factors can regulate neurite development cell autonomously or regulate the expression of cues and receptors from surrounding tissues. The complexity and dynamic nature of the nervous system makes in vivo analysis of regulatory mechanisms covering neurite development challenging. So we don't really have a good way to sort of get a precise measurement of these transcription factors. A lot of times they act combinatorically. So they're developing this means to sort of get at this signaling, this festival of signaling, as you could say. So it's hard to get a good account of the sort of the combinatorial nature of this transcription factor expression, these different programs. So that's what they're trying to do in this paper for C. elegans, because C. elegans is a relatively simple model. So animal models have been used to study neurite development, including mouse, rat, zebrafish, fruit fly, and C. elegans. This is prior work on neurite development itself. But what they're doing is they're developing this sort of map for the entire connectome. So here we review all published neurite development-related transcription factors in C. elegans. We've also collected and integrated bulk and single-cell transcriptome data to extract expression patterns of ligands, receptors, and transcription factors involved in neurite development. So they have the transcription factor data, which is a measurement of the RNA, the mRNA involved in this process. So you measure the abundance of the mRNA, and you can get a fix on how strongly a transcription factor is expressed in a certain cell in a certain part of the nervous system. Then they use GSoC data. So GSoC data is a way to measure epigenetic phenomena. So they're looking at how transcription factors bind upstream of genes, encoding different ligands and receptors involved in neurite development. So they actually look at how these genes are regulated with the GSoC data and then the abundance of mRNA in the transcription factor data. So you can get a sense of how these are being regulated by other genes and then how they're regulated locally in the connectome. And so they give a little overview of the neuronal wiring system. So neurites are neuronal projections that transmit information within the nervous system and to non-neuronal cells and tissues. Neurites are classified as axons and dendrites that transfer information from and to the cell body. In most vertebrates, axons are insulated by a protected myelin sheath that enables rapid electrical conductance, whereas some animals, such as C. elegans, lack such structures. So C. elegans doesn't have this myelin sheath like in other vertebrates. Depending on the neuron type, dendrites may be a single projection or highly arborized to receive information from multiple neurons. Neurites directly transmit information to target cells through synapses that release specific neurotransmitters or through electrical junctions that allow ion flow. So these are our chemical and electrical connections, respectively. And so neurite development is composed of projection, polarization, and extension of neurites that are controlled by dedicated signaling pathways and a cytoskeletal rearrangement. And so there are a bunch of reviews on neurite developmental processes and the mechanics and the anatomy of this. Mechanisms governing axon guidance in C. elegans have also been reviewed by Chisholm and colleagues. And this is this paper from 2016, which is here. This is the genetics of axon guidance and axon regeneration in C. elegans in the journal Genetics. So they've Figure 1 is this architecture of the C. elegans nervous system. So they show this, this is the amphid neuron, this is the pharynx, this is the nerve ring. So basically they show the schematic of the adult C. elegans hermaphrodite, and they show these major axodendritic processes. So you have your dendrites coming in from the sides of the organism, bringing in information from the sensory cells. You have this midline here that's bringing information up into the central nervous system here. So you can see all of these different structures that are involved in axodendritic processes. And so you can see that there are these projections that are needed throughout the body of C. elegans to bring information to different parts of the other parts of the body, including the central nervous system. So you can see where you need to have this process active across the organism throughout the extracellular matrix. B is a schematic of C. elegans hermaphrodite anterior nervous system. And you can see axons from the DNC, VNC, and amphid neurons where they enter the nerve ring, and they integrate information and govern behavior through these projections. So you have these different projections that come through. They converge upon the nerve ring. This nerve ring integrates information, and it's integrating information from the surface of the body, the pharynx, and the intestine. And so we'll take the FLP neurons for as an example. The FLP neurons have extensive dendritic arbors. That is what the PVD neurons enable mechano and thermosensation. And then in C, we have the schematic cross-section at the midbody showing these axonal tracts, in relation to other structures within the body. Their figure 2 shows neurite developmental regulatory mechanisms. So you have neurite development here where you have the differentiating neuron. You have from that cell body of these protrusions along the edge, which is looks kind of random, like it's kind of projecting in any direction it feels like. But then you have this next stage, which is a little bit more organized, which is engorgement, where you get these engorgements in different directions. So a set of different protrusions in a directed manner. And then you get consolidation, where you start to get these bundles of protrusions coming out from the cell body, and this is where they're consolidated into sort of the neuronal body that we're kind of more used to seeing in the adult stage. So you have these different stages of consolidation. This is stage 1 up here. Stage 1 is protrusion. Stage 2 is consolidation. Stage 3 is this sort of basic consolidation. Stage 4 is this more elaborate consolidation. And then stage 5 is where we start getting receptors and connections with neighbors. So these are your synapses here in green, these green and red caps at the end. And those serve as these chemical connections. Then in B, they show where we have this pioneer axon coming out. We have a developing axon by contrast, where the developing axon is sending out this protrusion, and there's this growth cone that is sort of at the end of the protrusion, and that allows for the axon to find its way through the extracellular matrix. So the axon is projecting out into the extracellular matrix. This growth cone is an area that senses the chemical concentrations, the chemical gradients, and allows for the axon to guide itself through that. And so the growth cone is guiding it through this milieu, and it's heading towards the target tissue, eventually making a connection with the target tissue. So it's the differentiation between sort of stage 3 and stage 5 of neurite development. So you can see here you have receptors on the end of this growth cone. They're getting information from these chemical signals and their concentrations. The microtubule polarity is moving outwards towards the growth cone from the cell body. This axon is expanding outward, and then it's trying to reach this target tissue. And if it doesn't reach the target tissue, the cell dies. But if it does reach the target tissue, then the cell can be electrically active and chemically active and live another day. And it successfully makes its target, say, with something like the muscle, or the muscle could impose physical restrictions on the distribution of these chemicals. And sometimes if you get something like muscle differentiating before the axon is reaching its target, it can actually guide it towards the target tissue. And then C is where we have this schematic of signals. So these signals are combinatorial. They're produced by the expression of a number of genes. So you get the expression here of UNC6, SLT1, UNC129, and MAB20. They have this combinatorial signal that forms this gradient. So they're the secreted signals. They form this gradient, the growth cones, which then follow these gradients. So the target tissue is sending out a combinatorial set of secreted signals. And likewise, the growth cone responds to a set of secreted signals. So UNC40, UNC5, LAD2, and MAG1 are all involved in that. And that's how they connect. So you have a set of genes involved in the growth cone, set of genes involved in the target tissue. And what they're talking about here with Neurite connectomes is largely the matching of these two signals so that the growth cone can make it to the target tissue properly. And of course, then the surrounding tissue excretes collagen 4, encoded by these three genes here, LET2, MP9, and MAD4. And this, of course, constrains this gradient so that it helps the growth cone along. Okay, so they talked about key ligands in the first part of the paper in the introduction. So they give these two tables where they kind of go over through the literature and they identify these different signals, and then they identify the genes that encode those signals. So you have different signal types, and then you have different genes that were identified by different references. So a lot of this work is a meta-analysis of the literature. So they've combined a meta-analysis with this data collection, and they're putting together this model. So as you can see, there are a number of signals here. We have ephren, TGF, SLIT, ECM, netrin, Wnt, and leukocyte cell-derived chemotaxin. And these are all kind of conserved across different neural systems. So you often see netrin and SLIT and ephren and Wnt throughout sort of animal neural development. So you'll see different versions of these signaling types across different nervous systems. And so you have, these are the genes in C. elegans is specified in the C. elegans genome. But of course you can find different homologs and paralogs in other genomes as well. Table 2 are membrane-bound proteins that control C. elegans neurite development. So you have these different receptor proteins, cadherins, and different other types of proteins. And then you have the genes that encode for these proteins. So for receptors, we have a number of genes that encode for receptor proteins, and those are reviewed in these papers. Cadherins, a number of genes encoding for cadherins that are cited in these papers, and so on and so forth. And then finally, table 3, we have transcription factors that control C. elegans neurite development. So this is where we have a developmental process, and that developmental process involves a number of different genes and their domains, and then we have they're expressed in a certain neuron type. So these graphs are interesting. These are graphs that show different genes for ligands and receptors, different genes for transcription factors, and then this pairwise correlation. For different in these first two, in A and B, for ligands and receptors and transcription factors respectively, you have these different time points from proliferating embryo to adult. So these are heat maps that just showed the level of upregulation for these different genes across the developmental period. So you see that there's a high degree of upregulation in the elongating embryo and the threefold embryo, which is post-gastrulation but before hatch. And you see there are a number of genes that are upregulated quite high, maybe around 5 to 10-fold. And then you have some genes that are upregulated before and after that period, up in this part of the heat map. So, you have to go back and look at the function of those genes to understand that a little bit better. But I like this where you have this distinction between sort of temporal upregulation and then gene-specific upregulation. And so you can see that pattern here. For transcription factors, we actually identify our genes along the side here by their family. And then correspondingly in B for transcription factors with the superfamily. So you kind of know what families and superfamilies these genes belong to. And then you look at the log or the upregulation here. So you see that there is a very high degree of upregulation in EGL46, which is a zinc finger. And I think we've seen papers before where zinc fingers are critical for certain processes. We also have other things that are not very much upregulated at all. So we have a number of cross-developmental stages, a number of genes where there's very little upregulation. And so that's kind of interesting as well. And then we have this parallelized correlation between candidate genes here, where you can see that there's this, so you're looking at the Pearson correlation. I think they misspelled Pearson here, where you get closer to 1 and there's a positive correlation. And then you also have negative correlations in some areas of this heat map, which means that they're not only sort of at random, but anti-correlated. And so there's a lot of information in this paper about the time course of gene expression, the time course of how these things are regulated. And we also have more information about, like this visualization here, where they have upregulation in this kind of spiral. And then they show the transcriptional regulatory network of ligands and receptors in figure 5. This shows these genes and how they're related in this network. And then we have the human orthologs of C. elegans transcription factors listed in this table. So this just shows the transcription factor gene in C. elegans, and then the human ortholog here. And then this kind of gives us a window into how to study disease through kind of the application of C. elegans. So we can actually look at some interesting transcription factor, look at its gene of origin, and then we can look at the human ortholog of that gene, and then we can look at if they cause a disease, and then we can get some information about the disease description and how maybe that information about development relates to human disease. So, C. elegans is used to study all sorts of conditions in humans, from different physiological functions to diseases. And one of the key components of that is that we can identify the orthologs that are involved. So the idea is that if a human ortholog is involved, or if we have something happening in C. elegans through the developmental time course, and this isn't necessarily something we can study in humans, we can go find the human ortholog and then see if we can replicate this in a more controlled study in humans or develop drugs with those gene targets in humans to see what happens. So this is all very interesting information as well. So one of the reasons why they looked at this sort of list of genes and they built all these heat maps and everything is that combinatorial gene regulation requires the activity of multiple transcription factors. So they had to do this kind of looking at these heat maps and then looking at these networks. One of the reasons why is because genes don't act in isolation, they act in these sort of networks, and then they act together to form this combinatorial signal. Another interesting point here is that by kind of building this combinatorial gene regulatory model, they can identify not only the conditions for growth cone persistence and the proper axonal guidance, but they can also identify maintenance factors, which are involved in plasticity and axon growth following injury. So, this is again important for the human domain, but also understanding plasticity more generally. So finally, the dynamic and combinatorial behavior of neurite development regulators is reflected in the transcriptome, particularly from single-cell level analysis. This information may shed light on how to manipulate these regulators to overcome neuronal deficits and neurodegenerative decline. So that's all for today. Hopefully you found this useful, and see you next week.